In this video, I'm going to be talking about like a long sequence of how to effectively and accurately target cancer to eradicate it. So the first step is the lipid transport, usually with macadamia nuts, because macadamia nuts have healthy fats that actually help enhance these things called kilomicrons. The kilomicrons are needed to efficiently deliver fat-soluble vitamins such as A, D, E, and K to immune cells and stromal cells. So if the kilomicrons aren't working, then the fat-soluble vitamins won't be delivered. So macadamia nuts are rich in oleic acid and profiles, and this actually avoids CD36 oxidized LDL inflammatory signaling. So as a result of the fats being absorbed, the vitamin D receptor works, so VDR for short. The vitamin D receptor is a zinc finger nuclear receptor. So basically this means that zinc won't work if the vitamin D receptor is not working, or if it's not stimulated. With enough zinc, with adequate zinc and vitamin D, the closed chromatin at oncogenic promoters will close. So that means that if the chromatin is closed and the DNA is inside of the chromatin, that oncogenes won't work. If oncogenes aren't working, then cancer cells won't work, or at least will be weakened. This will only work if the chromatin is closed. Now, if the chromatin is opened, then that means that the cancer cells will flourish because the oncogenic promoters are working. So this supports cytotoxic lymphocyte maturation and reduced pro-tumor NF-κB tone. NF-κB is it's an inflammatory mediator cancer cells have to survive. Also, the macadamia nuts have arginine, and this counters arginine depletion by tumor arginase, because in the tumor microenvironment actually hogs arginine for energy, and that can be replenished with macadamia nuts. As a result of the arginine replenishment, this preserves nitric oxide production in T cells and macrophages. Now, a good way to open chromatin is with sulforaphane, which is in vegetables like Brussels sprouts, broccoli, cauliflower, kale. Sulforaphane is a histone deacetylase inhibitor. This causes chromatin to relax, but not indiscriminately. And this biases transcription toward antioxidant genes and cytotoxic effector genes when immune activation signals are present. So that pretty much means that with the chromatin opening, the transcription factors for antioxidant genes are working. Because if they're not working, then the antioxidant genes won't work, and then the antioxidants won't work, which means that there's going to be a lot more reactive oxygen species. So the goal of having cruciferous vegetables is to reduce those reactive oxygen species. So basically, sulforaphane doesn't activate oncogenes. It actually, oncogenes require growth factor signaling and NYCRAS, which is like technical jargon. But in immune cells, sulforaphane actually enhances granzyme B and perforin. The immune cell attaches to the cancer cell with perforin, and then in the perforin, the granzyme B goes in through the perforin and then activates apoptosis in the cancer cell. Also, sulforaphane also enhances antigen processing machinery. So basically, that means that antigen presentation is working, and if that's working, then the immune cell can see the cancer cell. Because if antigen presentation isn't working, then the immune cell won't be able to see the cancer cell, and the cancer cell will actually escape immune surveillance. Okay, so another piece here is vitamin B6, which helps magnesium works, which will help metal handling, which will increase mitochondrial ATP, because magnesium is required for ATP synthase and kinase signaling, through AMPK and mTOR balance. This stabilizes DNA-protein interactions for transcription fidelity. Vitamin B6 enables amino acid metabolism and heme synthesis. So heme synthesis is another word for iron synthesis. So copper and iron transport enables the proper handling of functional ETC complexes. This supports sustained ATP in immune cells and prevents iron sequestration that tumors exploit. So basically, vitamin B6 enables heme synthesis that enables copper and iron transport, because if vitamin B6 is missing, the iron will actually get sequestered in macrophages that the tumors exploit. If the iron is sequestered, that means that the mitochondria will not work, because the mitochondria has to have iron to work. So if the iron is sequestered in macrophage, then the mitochondria is not going to have the iron that it needs, which means that the mitochondria will not make ATP. If there is no ATP, then the immune cells won't work, because immune cells need ATP to function. Immune cells are a high energy demanding cell, and without enough energy, they can't attack the cancer cells. The piece here is vitamin A and vitamin B12, which is redox control for immune signaling. So vitamin A, or another way to say it is retinoic acid, enhances antigen presentation and immune cell homing. Again, antigen presentation is needed for the immune cell to be able to see the cancer cell, so that the immune cell can attack the cancer cell. So vitamin A is in sweet potatoes. You can find vitamin A in carrots, which is one of my favorites. Vitamin A also supports calreticulin exposure synergy. So basically CRT for short. CRT is on the cancer cell. If CRT is on the cancer cell, the CD91 on the macrophage can bind onto the CRT on the cancer cell and then promote apoptosis or phagocytosis. So, and then vitamin B12 maintains methylation balance, which supports glutathione regeneration. As a result of vitamin B12 making glutathione work, this makes cysteine thiol integrity work. What cysteine does, cysteine thiols or residues does, is that it weakens microbial and tumor-associated biofilms, which improves immune penetration. As a result of cysteine weakening the biofilm, the immune cells can actually attack the cancer cell. The next piece is eat me signals for phagocytosis, which can actually be enhanced by oranges, because oranges has vitamin C. This promotes the translocation of calreticulin, or CRT for short, on the tumor surface, so that the CRT goes to the tumor surface instead of hiding inside the tumor. Because if the CRT is hiding in the tumor, then the CD91 on the macrophage can't bind onto the CRT. If the CD91 can't do this, then the macrophage can't make the tumor promote apoptosis or phagocytosis, which is the immune cell engulfing the tumor. So the only way for that to work is for the CD91 on the macrophage to bind on the CRT on the tumor. The CRT has to be on the surface of the tumor. So the CRT might be hiding in the tumor. So this is where oranges helps, because oranges has vitamin C and flavonoids. Another piece is ginger for innate immune recalibration. Ginger has gingerols and shogols. This blocks toll-like receptor overactivation on macrophages. As a result, this reduces immune exhaustion and better antigen presentation. Because if toll-like receptor is overactivated by, let's say gluten creates lipopolysaccharides that actually overactivates toll-like receptor 4. This means that the immune cell won't be able to see the cancer cell, and then the immune cell can't attack the cancer cell. So in order to block this, the ginger blocks the overactivation of toll-like receptor 4 so that the immune cell can see the cancer cell. Next piece is a capsaicin, which is in like chili, red chili. This actually starves tumors of being able to be metabolically active. So capsaicin activates brown fat thermogenesis. This increases fatty acid oxidation, which increases nutrient diversion because brown fat can actually hog nutrients and the glucose away from tumors so that the tumor can't grow because cancer tumors need glucose to grow. So the next piece is resveratrol. Resveratrol activates SIRT1. SIRT1 activates PGC1A, which actually enhances mitochondria. If a mitochondria is enhanced, then this creates more ATP for immune cells because immune cells need ATP to work. But PGC1A also suppresses CD36, tumor cells used to avoid immune detection. Consequences are improved immune endurance, reduced reliance on CD36, and blunted oxidized LDL inflammatory signaling. Next piece is a bromelain, which is needed for physical access to the tumor. So bromelain cleaves CD44, CD62L. This allows the immune cell to physically access the tumor so that the immune cell can attack the tumor. So bromelain, which is in pineapple, improves immune cell trafficking and tumor microenvironment infiltration, reduced physical immune exclusion. So if the tumor microenvironment is opened, then the immune cell can go in. Okay, as the immune cells go in, they're going to be attacked by cytokine inflammatory attack from tumors, from cancer tumors, because the cancer cells can actually defend themselves from tumor attack. Immune cells can use astaxanthin as a shield to protect from a tumor-derived cytokine attack. But you can find astaxanthin in salmon, which you can find at the store. Next piece is the arginine, which can be found in macadamia nuts. Arginine boosts nitric oxide, which actually directly attacks the tumor. So that is a direct tumor cytotoxicity. Nitric oxide also enhances calreticulin exposure. Like I said earlier, CRT for short. The CRT is in the tumor or in the cancer cell, and the nitric oxide brings the CRT out to the surface of the tumor instead of hiding. This way, the CD91 on the immune cell can bind to the CRT and promote phagocytosis. So recap, the chromatin is opens with sulforaphane, like in Brussels sprouts, cauliflower, broccoli. High mitochondrial ATP in immune cells with oranges. CRT, which is short for calreticulin, on cancer cells is exposed, and then the CD91 on macrophages binds onto the CRT to promote phagocytosis. Improved antigen presentation by reducing toll-like receptor 4 activation that are exposed on macrophages. Toll-like receptor activation is activated by lipopolysaccharides. So to block that activation, use ginger. Recap: reduced CD36, oxidized LDL inflammatory signaling. Another recap piece is enhanced immune infiltration with the bromelain, cleaving of CD44-62L, which is you can find in pineapple. Metabolic starvation of tumors with brown fat. Arginine from macadamia nuts to boost nitric oxide mediated tumor stress. So nitric oxide attacks the tumor. Nitric oxide also enhances calreticulin exposure of CRT on the outside of the tumor instead of calreticulin CRT hiding inside of the tumor. Because if that calreticulin is hiding in the tumor, then the CD91 on the macrophage can't bind onto the tumor. Make sure to subscribe, leave a like, and I'll see you in the next video.